morning, everybody. How are you all doing? I hope you are extremely excited, fully focused, and very, very dedicated towards your studies and making sure that every day, every second, and every minute is counting when it comes to your time, and you're making sure that you are studying day in and day out to achieve your dreams. Yes, so to add more fire to your dreams, to make sure that you do not ever give up on your dreams, I am going to start today's class by talking about two extremely dedicated children. Yes, two phenomenal students who have not allowed anything to come in between they and their dreams. Let us start today's class with this great boy whose name is Prince Kumar. Prince Kumar's father drives a bus for the Delhi Transport Corporation. Yes, and you know his salary is extremely limited. They had a small washroom in their house. Yes, they had a small bathroom in their house, which they converted into a study room for their children. Yes, and Prince and his sister took turns to study there. Are you getting my point? And when both of them had to study, one of them used to sit in the small room and study, one of them used to go to the converted washroom to study, and both the parents used to sit quietly in the kitchen. ऐसे होती है मेहनत, ऐसे होती है पढ़ाई, जी जान एक करना पड़ता है, रात दिन एक करना पड़ता है, तब जाकर रिजल्ट मिलता है. क्या आप लोग लगे हुए हो जी जान और रात दिन एक करने में? Yes, everybody, this boy is a Delhi government school topper of class 12th. Understood? Yes, and he wants to join the National Defense Academy or also wants to clear the IIT examination. But इस बच्चे को ट्यूबरकुलोसिस हो गया. इनके पास इतना पैसा भी नहीं था कि ये लोग उसको अच्छा फल फ्रूट खाना खिला सके ताकि वो जल्दी से ठीक हो जाए. But still, this boy topped. Is that clear? This girl is from Ahmedabad and her name is Afreen. She is one of the 4 children that her parents have. Her father, as you can see from this photograph, is a simple auto driver. But this girl's dreams are very big. सपने कुछ देखकर नहीं आते हैं, सपने सिर्फ आते हैं और उनको फिर पूरा करने के लिए रात दिन एक करना पड़ता है. Yes, my dear children. So this girl has topped class 10th. She wants to become a doctor just like you all, and she's working very hard in her limited means to achieve it. So आज से I don't want any excuses. If these kids can do it, so can all of you. Yes, I don't want any excuses. I want you to study morning, afternoon, evening, night, every day. Study as much as you can and make sure that you achieve your dream. Done? Yes. So no excuses, no telling that we can't do this and we can't do this. You are just simply doing it. So moving ahead with today's lecture, my dear children, which is on an extremely important topic from where a lot of MCQs comes, is biotechnology. Cool. So today we are going to do biotechnology. About the theory of biotechnology, we have 3 lectures which are planned for biotechnology. About the theory of biotechnology, I have taken 2 webinars on biotechnology. Yes, we have already done 2 webinars on biotechnology. There are some YouTube lectures also which are available. Please do them in detail. I got a mail from a student yesterday that child had seen all of my molecular basis of genetics MCQs and can solve all AIIMS level everything questions. So please see the webinars and the YouTube lectures. Everything has been done in detail. See them. Come prepared every day. We are going to do a series of 3 lectures on biotechnology. Today is the first lecture of biotechnology from the 3-lecture, 3-hour series. Come prepared with doubts. Ask me your doubts during this time. Leave your doubts behind in the comment section. I will make sure that today I will sit. Today and tomorrow I will simply sit and answer all your doubts on YouTube. Cool? Chaliye. So let's start. Extremely cool, extremely important, simple, nice, important class on biotechnology. Yes, I have done agarose gel electrophoresis in detail. आप सबको पता है जितने बच्चों को मैंने बायोटेक्नोलॉजी के YouTube लेक्चर में पढ़ाया है, we have done agarose gel electrophoresis. You remember that? एवरीबडी यस। आज देखिए अगरोस जेल इलेक्ट्रोफोरेसिस से कैसे डायरेक्ट सिंपल क्वेश्चंस आए हुए हैं। सो कम लेट्स स्टार्ट। क्वेश्चन नंबर 1 ऑन योर स्क्रीन, अगरोस जेल इलेक्ट्रोफोरेसिस से ही आ गया। यस, जेल इलेक्ट्रोफोरेसिस, बाय व्हिच आई मीन agarose gel electrophoresis, is used for. Why do we use agarose gel electrophoresis? Do we use it to construct a recombinant DNA? Is it used for isolation of DNA molecules? Is it used to cut the DNA into small fragments? Or is it used to separate the DNA molecules according to their size? Come on. Yes, absolutely right. It is used for separation. Let me explain this in detail. Agarose gel electrophoresis is a method of separation the DNA molecules according to their charge. Yes, so what is the charge separation? हम एक ऐसे प्लेट ले लेते हैं, इस प्लेट पे हम agarose gel डाल लेते हैं, which we get from agar, right? Soft gel so that DNA is not damaged. DNA is negatively charged, right? Because the DNA is negatively charged, it will move towards a positively charged anode. Right? When I was your class, when I was in class 10th 11th, मुझे एनोड कैथोड याद करने में बड़ी प्रॉब्लम होती थी. सो एनोड ऐसे लिखते हैं, ए ऐसे बनाते हैं. सो आई नो दैट एनोड इज पॉजिटिवली चार्ज्ड. How do we make an anode? Like this. So A, yes, positively charged anode. So हम जब DNA इधर डालेंगे, DNA will move towards the anode. Now we cut the DNA into fragments. Let me show you. You see how I've cut the DNA. So can you see there are 3 fragments here? Let us name them. Fragment number 1, fragment number 2, and let's call this fragment number 3. Can you all see that fragment number 3 is the smallest? So because fragment number 3 is the smallest, it will also be the lightest. Right? It will be the lightest. Molecular weight will be the lightest of fragment number 3 because it is the smallest. So, very simple. When I put it here, when I put this here, the smallest fragment will go here because the smallest fragment will have a low molecular weight. So small will have low molecular weight, therefore it will move ahead. So with the help of agarose gel electrophoresis, I am going to separate the DNA fragments according to their size. Now I want to see whether the DNA fragments are moving or not, which fragment is moving ahead, which fragment is staying behind. So I will use a dye. The name of the dye is known as ethidium bromide. What is the name of the dye? Correct, ethidium bromide. And when I add ethidium bromide to this mixture and I see it under ultraviolet radiation, I am able to see orange colored DNA fragments. So orange colored DNA fragments are seen in the agarose gel electrophoresis plate under the dye ethidium bromide when you see it under a ultraviolet rays. So what is agarose gel electrophoresis? It is a method of separation of DNA fragments. How do you cut the DNA fragments? You cut the DNA fragments with the help of restriction endonuclease and you separate them by agarose gel electrophoresis. The smallest is the lightest. Right? I repeat, the smallest is the lightest, so it moves ahead and we are able to see it. Very simple. Any doubts? 100% a question will come from agarose gel electrophoresis. These fragments can be separated. The cut DNA fragments, which I cut with the help of restriction endonuclease, this is the enzyme known as molecular scissors. I cut it with the help of this enzyme. And the cut fragments are separated by a technique known as gel electrophoresis. DNA are negatively charged. They move towards the anode. They move according to their size. Yes, so the smallest ones, the smaller will move the farthest away. I cut it and I remove it from the gel. How do I see it? I see it with the help of ethidium bromide dye under UV radiations. And then whatever material I want, I have been able to isolate it and use it in whichever way I want. Is that clear? This is a theory chart which has everything mentioned about agarose gel electrophoresis, which can be asked in your MCQ questions. Next question on your screen now. Polyethylene glycol. For what is it used? Yes, polyethylene glycol is used in plants, or especially when you're using anything to clone in plants to do the gene transfer without a vector. We have done this word at least 2 to 3 times in YouTube lectures only. Only and only if you follow YouTube lectures, you will be able to do well in your board exams or your NEET exams. Yes, board exams तो हो गए आप लोगों के. Correct? Even in your NEET exams. Direct gene transfer is the transfer of naked DNA, that means without covering, into plant cells. But there is a rigid plant cell wall made up of, yes, cellulose, hemicellulose, and pectin, right, which is a barrier which will not allow the uptake. Therefore, protoplasts are the favored target for gene transfer. What is protoplast? The inner material, the cytoplasm and the nucleoplasm. That's where I want to transfer. Polyethylene glycol mediated DNA uptake is direct gene transfer medium that utilizes the interaction between polyethylene glycol and naked DNA. So ethylene glycol will loosen up the DNA so that they can attach to each other. What is a vector? A vector is any material, anything which carries the DNA into the host cell. अगर मुझे vector के बिना carry करना है, I don't need a plasmid, I don't need a cosmid, I don't need a lambda phage. मुझे उसके बिना transfer करना है in the DNA. I will use polyethylene glycol. So polyethylene glycol will cause vectorless transport. Right, it will transport the DNA into cytoplasm so that cytoplasm में जाके वो दूसरे DNA के साथ fuse करके एक recombinant DNA बन जाए. But this happens without a vector. Cool. Moving on to the next one. Bioreactors हमने कल भी किया है. We did sewage tanks which are, which have the similar kind of MCQs which come on bioreactors. Yes, everybody. Stirred tank bioreactor. That means bioreactor is anything जहाँ पे बहुत large amount में vaccines produce होती है. बहुत large amount में recombinant DNA technology का काम होता है. That is known as a bioreactor. So जब मैं उसको stir करती रहती हूँ, that means I have a big bioreactor like this. ध्यान से देखिए. And there is a stirrer. So I'm going to use the stirrer, absolutely everybody, and keep stirring the contents of the bioreactor. So they are asking you that why do we need a stirred tank bioreactor? Is it for adding preservatives? Is it for purification of the product? Is it to ensure anaerobic conditions in the vessel? क्या ये सारा oxygen खींच लेगा बाहर? Or is it for availability of oxygen throughout the process? Yes, it is for availability of the oxygen because oxygen usually release किया जाता है one end से. So when it continues to revolve, it will mix everything and it will make sure that oxygen हर जगह पहुंच जाए. Is that clear? So stirred tank bioreactors have been designed for the availability of oxygen throughout the process. Yes? Done? Okay. So you see, this is the stirrer. Can you see that, everybody? This is the stirrer. A stirred tank bioreactor is usually cylindrical or with a curved base to facilitate the mixing of the reaction contents. Right? Curved base, ताकि हम उसमें, गोल गोल घुमाकर इसको हम mix कर सके. It's like a churner. Right? It's like a mixer grinder. The stirrer facilitates mixing and oxygen availability throughout the bioreactor. Even mixing means बराबर से सब कुछ mix हो जाना चाहिए. Alternatively, air के bubbles भी release कर सकते हैं. If there is aerobic bacteria, they need air. So alternatively, I can even release the bubbles of air. Cool? Any doubts? Also, my dear children, I really want to tell you, see, the last 22-23 days are remaining. आपको जो भी डाउट आ रहा है, you write it down in the comment section. उसके अलावा, you need specifically some topic. We will do it. We will make small modules on those small topics. आपको specifically कुछ चाहिए. Any topic which you all find is very, very difficult, write it down. You need to tell us. You need to be heard. You need to tell us कि हमें ये problem आ रही है. Write it down in the comment section. We will respond back. Yes? Okay. In genetic engineering, a DNA segment or gene of interest जो मुझे चाहिए is transferred to the host cell. Host cell वो होगा like E. coli. राइट तो जीन ऑफ इंटरेस्ट क्या होगा मेरे को इंसुलिन बनाना है मुझे आर्टिफिशियल इंसुलिन बनाना है जिसको मैं यूमुलिन कहती हूँ सो जीन ऑफ इंटरेस्ट क्या होगा जीन ऑफ इंसुलिन व्हिच आई एम गोइंग टू ऐड इन द यस in the E. coli. So insulin is my gene of interest. Yes, what is my gene of interest? Insulin. Correct? And how do I transfer this? I transfer this with the help of a vector. Let us call this plasmid. Consider the following four agents. बैक्टीरियम, प्लाज्मिड, प्लाज्मोडियम, बैक्टीरियोफेज, इनमें से कौन सा यूज कर सकते हैं एज अ वेक्टर, एज अ कैरियर? Very simple question. I'm sure everybody, yes, you've all answered that. Extremely simple question. Only plasmid or bacteriophage. Bacteria cannot be used as a vector. No, it can be a plasmid, right? Plasmid ये बैक्टीरिया का डीएनए है, ये बैक्टीरिया का डीएनए है. प्लास्मिड इज एक्स्ट्रा क्रोमोसोमल, क्रोमोसोम के अलावा, लेट मी राइट इट डाउन, प्लास्मिड इज एक्स्ट्रा क्रोमोसोमल, राइट, एंड इट इज ऑलवेज डबल स्ट्रैंडेड सर्कुलर पार्ट. इसके अंदर हम फॉरेन जीन, लेट अस से दिस इज इंसुलिन का जीन, सो इनसाइड द प्लास्मिड, प्लास्मिड इज एक्स्ट्रा क्रोमोसोमल डबल स्ट्रैंडेड सर्कुलर डीएनए व्हिच इज प्रेजेंट इन बैक्टीरिया. आई रिपीट, plasmid is extra chromosomal double stranded circular DNA. I have taken webinars, I have taken YouTube lectures of plasmid in detail, where I've done antibiotic resistance gene also in detail. Please see them, please. Right? So bacterium, yeah, correct everybody. So bacterium नहीं होगा, plasmid होगा, plasmodium तो मलेरिया कॉज करता है, ये भी नहीं होगा, and bacteriophage like the lambdaphage. Correct? Viruses. Also remember this, the virus can transmit information from one bacteria to the other, right? Information or DNA or genetic information can be transferred from one bacteria to the other with the help of virus. Such transmission of information is known as transduction. What is it called? Transduction. There are three methods of information transfer. Please listen to me carefully. What are the three methods of information transfer? The first method of information transfer, which happens from one bacteria to other in the external medium. Yes, in the external. Let me explain this to you, please. Right? So, ये रहा एक bacteria. Let's call this bacteria A. ये दूसरे bacteria को let's call bacteria B को information transfer कर रहा है through the external medium. This is known as transformation. This was discovered in streptococci pneumonia by a scientist whose name was Frederick Griffith. Very important from MCQ point of view. He called the information which is going and which is changing the bacteria B as transforming principle. Also, एक बैक्टीरिया A है, ये दूसरे बैक्टीरिया B को इंफॉर्मेशन दे सकता है with the help of this stick known as pilus. Right? So this bacteria is F positive, जिसको information दे रहा है F negative. This passage of information through the pilus is known as conjugation, or information can go from bacteria A to bacteria B with the help of a virus. This transformation of information is called as transduction. What is this called? Transduction. Virus, very important. जिस भी bacteria को infect करेगा. So there is transformation, write it down. There is conjugation and there is transduction. Very important from MCQ point of view. These are three methods of sexual transmission of information in a bacteria. Now, virus, this is a bacteria. This is a virus. The virus is going to अपना डीएनए, देखिए ये इसका डीएनए, ये इसका डीएनए इसमें इंजेक्ट कर देगा. दिस इज द होस्ट. लेट अस कॉल दिस द बैक्टीरिया, जो होस्ट है. सो जब एक बार वायरल डीएनए इसके अंदर आ गया, वायरल डीएनए एक बार होस्ट में आ गया, द होस्ट हैज टू डाई. I will discuss this in more detail in subsequent classes also. Very important. Host has to die. अगर viral DNA immediately host को destroy कर देता है, ये host को destroy कर देता है immediately. This is called the lytic life cycle. अगर वायरल डीएनए थोड़ी देर तक होस्ट के अंदर रहता है और होस्ट को डिस्ट्रॉय करने के लिए थोड़ा सा टाइम लेता है, आई रिपीट, इफ द वायरल डीएनए स्टेज इनसाइड द होस्ट एंड टेक्स लिटिल टाइम, I repeat, if the viral DNA stays inside the host and takes little time to destroy the host, it is called as lysogenic life cycle. So it takes some time, lysogenic life cycle, it immediately destroys, lytic life cycle. Any doubts? So plasmid and bacteriophage are used as vectors in genetic engineering. Plasmid अपने आप replicate करता है. It is not dependent on anybody. Autonomous है, extra chromosomal है, found in bacteria. right? Plasmid is very important because plasmid has got antibiotic resistance gene. The antibiotic resistance gene will help us in biotechnology to find out कि कौन सा बैक्टीरिया ट्रांसफॉर्मेंट है, कौन से बैक्टीरिया ने फॉरेन जीन ले ली है, कौन सा बैक्टीरिया ट्रांसफॉर्मेंट है और कौन सा बैक्टीरिया नॉन ट्रांसफॉर्मेंट है. Is that clear? Bacteriophage is a virus. It lives within the host. It destroys the DNA of the host. If the virus destroys the DNA of the host immediately, lytic life cycle. If lysis, lytic, if the virus takes some time to destroy the DNA of the host, lysogenic life cycle. Every word that I'm saying is important. Every word, right? So plasmid is extra chromosomal genetic material. It contains antibiotic resistance gene. It is important because it will tell you which is transformant, which is non-transformant. Is that clear? Cool. Cool. Chaliye. Next question. There is a restriction endonuclease. What is restriction endonuclease? It's going to cut the plasmid called EcoR1, right? What does co mean? E stands for Escherichia, right? Co stands for coli. R is the strain, right? R is the strain. कई बैक्टीरिया बहुत ज्यादा डिजीज कॉज करते हैं, विरुलेंट. कई बैक्टीरिया इतनी डिजीज कॉज नहीं करते, ए विरुलेंट. सो हमें पता होना चाहिए whether it is a virulent or a virulent bacteria. One is because it was the first enzyme to be isolated from Escherichia coli. So E is the genus and CO is E. coli, correct? Is that clear? E. Co. Co stands here for coli, like at this written. The first letter for the enzyme is the first letter of the genus name in italics of the bacterium. Then comes the first two letters of its species. Next is the strain. At last is the Roman numerical signifying the order of discovery. So co are, yes, our strain, and it was first endonuclease to be isolated from Escherichia. अगर आप लोग में से कोई सुपर कूल साइंटिस्ट बनता है और इश्चरीशिया कोलाई से कोई फिफ्थ एंजाइम ढूंढ निकालता है तो वो हो जाएगा इको R5, राइट? सो इश्चरीशिया कोलाई, correct. R strain, correct. And it is the first enzyme to be isolated, so it will be EcoR1. This is how Escherichia coli looks. It has a tuft of flagella at one end. It's a very, why this thing here? It's a very common, residing bacteria of the intestine, extremely commonly found in intestine. So this is the nomenclature of the enzymes: Escherichia coli, R strain, first enzyme to be isolated. Cool. We should move ahead now. Next question on your screen now. Yes, PCR, polymerase chain reaction. Polymerase chain reaction was discovered by a scientist whose name was Carey Mullis. क्या नाम था scientist का? This can come in MCQ. Carey Mullis. What does PCR do? PCR helps us to amplify the amount of DNA. जितना मेरे पास DNA है, अगर उस DNA को मुझे बढ़ा चढ़ा कर दिखाना है, I have to amplify the amount of DNA. What do? I do a polymerase chain reaction. So polymerase chain reaction and restriction fragment length polymorphism. Restriction fragment length polymorphism are RFLP, right? Are the methods for doing what? They are the methods, exactly, well answered. They are the methods to do genetic fingerprinting. Now, let me explain this. आप एक criminal forensic expert बन गए. Yes? और आप एक मर्डर की साइट पे जाते हैं और उस मर्डर की साइट पे आपको एक ड्रॉप ऑफ सलाइवा मिलता है, सिर्फ एक ड्रॉप ऑफ सलाइवा, या आपको सिर्फ बाल, वन सिंगल हेयर स्ट्रैंड मिलता है, राइट? सो टेल मी, क्या इसमें से बहुत ज्यादा डीएनए निकलेगा? Will you be able to take out like 1 quintal ton of DNA out of this? No, it will be very little DNA you will get from here. Exactly. Out of this very little DNA, क्या आप बहुत सारे टेस्ट कर सकते हो? Can you do many tests? No, you will not be able to do so many tests. So is it nice to have increased DNA so that you can do all tests? Is it better if you can increase the DNA? So you will do a test called PCR, which is meant for amplification of DNA, which is meant to increase the DNA, which is meant to amplify, increase the amount of DNA. And when you have amplified the DNA, then you can do all tests and do DNA fingerprinting. Now tell me, do each one of us have a unique DNA? Each one of us has a unique DNA which does not match with anybody. That uniqueness of a DNA is known as, please pay attention, that uniqueness of a DNA is known as VNTR, variable number tandem repeat, right? It is the one which helps us to do DNA fingerprinting. So that poly means different, morph means appearance. Each one of us has a different type of VNTR. Right? I repeat, each one of us has a different type of VNTR, which is not similar, not same as anybody else. That is called as VNTR, restriction fragment length polymorphism, which helps us to identify ye kiska DNA hai, which is known as, yes, DNA fingerprinting. Polymerase chain reaction is used to amplify, means increase. Amplifier is there in your speakers, which is used to increase the sound of the music, which is going to increase a small DNA fragment into a very large quantity. PCR is very helpful in DNA fingerprinting, where we get very small blood, very small hair, very small drop of saliva from the crime scene. Right? Now, RFLP, restriction fragment length polymorphism, corresponds to the occurrence of different cleavage sites for restriction enzymes in the DNA of different individuals. मेरी बॉडी में डीएनए कट होने के लिए एक रिस्ट्रिक्शन साइट, व्हाट इज रिस्ट्रिक्शन साइट? इट्स अ पैलिंड्रोम, right? Restriction site is a palindrome, GAATTC, GAATTC like this, GAATTC like this. For example, madam, MADAM like this, MADM like this. So each one of us have different restriction sites, right? हमारी बॉडी में अलग अलग जगह पर रिस्ट्रिक्शन साइट्स होंगी. दे आर वेरी वेरी इंपॉर्टेंट जेनेटिक मार्कर्स. अलग अलग VNTRs होंगे, अलग अलग जगह रिस्ट्रिक्शन साइट्स होंगी, जिसकी वजह से हम हर किसी को एक इंडिविजुअल के तौर पर देख सकते हैं, which are very important for gene mapping and gene tracking. It's used in DNA fingerprinting. Each one of us has a unique restriction sites. Which one of the following is wrongly matched? Come on. Yes? Do you use the vector DNA to synthesize transfer RNA, or is it used to carry the foreign gene? Tell me. Is it used to carry the foreign gene, or is it used to make the transfer RNA? That reminds me, please see translation. The translation 1 hour video of YouTube, from that 100% MCQs will come. 100%. Micropropagation में हम plants वगैरह उगाते हैं laboratory में, yes. The unorganized mass of cell, this I've done in yesterday's class. Unorganized mass of cell, जिसको मैं ऑक्सिन साइटोकाइनिन देने वाली हूँ, is called callus. When I fuse two different cells, it's called a somatic hybridization. But vector DNA is to carry foreign substance. Vector DNA is not meant to carry or synthesize tRNA. This is definitely a wrong statement. Yes, vector DNA are molecules that carry foreign substances. They are not meant for tRNA. Yes, they are not meant for peptide synthesis. वेक्टर डीएनए का कोई लेना देना नहीं है इससे, मैसेंजर आरएनए से कोई लेना देना नहीं है, टीआरएनए से कोई देना. वेक्टर डीएनए is for recombinant DNA technology. Cool? Any doubts? Anything that you have not been able to understand, anybody? Please write down in the comment section, the comments of the normal class. Right? I will respond back to you. In genetic engineering, the antibiotics are used as what I just told you. हर plasmid में एक antibiotic synthesizing site होगी. Right? So ये plasmid है, there will be a site for antibiotic resistance. So tell me, if it, अगर किसी बैक्टीरिया के पास plasmid है, अगर किसी बैक्टीरिया के पास plasmid है with antibiotic resistance, right? Very simple, pay attention. एक और बैक्टीरिया, उसमें हम बनाते ही नहीं है plasmid. There is no plasmid, no antibiotic resistance, nothing. इसके पास है. Can it take the foreign DNA? ये foreign DNA मान लीजिए. This is the foreign DNA. Let me draw it here. तो अगर प्लास्मिड इसमें फॉरेन डीएनए लग सकता है, yes. इसमें प्लास्मिड ही नहीं है, तो क्या फॉरेन डीएनए लगेगा? There is no plasmid, there is no foreign DNA. Correct? इसके पास प्लास्मिड है, एंटीबायोटिक रेजिस्टेंस साइट तो प्लास्मिड में होगी ही, तो इसमें फॉरेन डीएनए भी आकर लग जाएगा. So, can the antibiotic resistance tell you कि फॉरेन डीएनए कहाँ लगेगा? अगर यहाँ फॉरेन प्लास्मिड ही नहीं है, एंटीबायोटिक रेजिस्टेंस साइट ही नहीं है, तो फॉरेन डीएनए लगा ही नहीं. सो वी डोंट वांट टू ग्रो दिस. मुझे इसको कल्चर मीडियम में ग्रो नहीं करना है. Is that clear? So is it going to tell me, is it going to act as a selectable marker and tell me जिसके पास foreign DNA लग जाता है, that is known as, absolutely right, that is known as a transformant. जिसके पास foreign DNA नहीं होता है, that is known as, नहीं होता है, नहीं होता है, that is known as, exactly, that is known as a non-transformant. What is it called? Non-transformant. Yes. So is this going to help us to find out which is a transformant and which is a non-transformant? Is genetic engineering का antibiotics, because antibiotics कहाँ लगेंगी जाके? Plasmid में. Absolutely. So is it going to tell us that which is a transformant and which is a non-transformant? So it's a marker. I don't want the non-transformants, I want the transformants. Correct? Cool. Selectable marker are those genes which help in selecting those hosts which contain vectors, that is transformants, and eliminating the non-transformants. ये antibiotic resistance, देखो ये plasmid है, ये plasmid बनाया मैंने, राइट. ये रहा आपका सिलेक्टेबल मार्कर. मार्कर मतलब ये एक मार्क है. किस चीज का मार्क? एंटीबायोटिक रेजिस्टेंस का. किसका? एंटीबायोटिक रेजिस्टेंस का. और ये है मेरा जीन ऑफ इंटरेस्ट. तो अगर प्लास्मिड है, उसके अंदर सिलेक्टेबल मार्कर है, तो जीन ऑफ इंटरेस्ट लग जाएगी. अगर प्लास्मिड नहीं है, सिलेक्टेबल मार्कर नहीं है, तो जीन ऑफ इंटरेस्ट नहीं लगेगी. सो can I find out with the help of selectable marker कहाँ जीन ऑफ इंटरेस्ट लगेगी? So can I call this transformant? ये ट्रांसफॉर्म हो जाएंगे, जीन ऑफ इंटरेस्ट ले लेंगे. And can I call them non transformant? ये ट्रांसफॉर्म नहीं होंगे, जीन ऑफ इंटरेस्ट नहीं लेंगे. Is that clear? So there are antibiotics like ampicillin, there is antibiotic like tetracycline. Correct. In सब में selectable markers होते हैं. Antibiotic resistance are selectable markers which help us to identify the transformants from the non-transformants. So tetracycline resistance or ampicillin resistance antibiotic markers हैं which very easily help us to find out which are transformants and which of them are non-transformants. Cool. चलिए, which one of the following represents a palindromic sequence? चलिए बताइए इनमें से कौन मलयालम के जैसा है? Yes, that means Malayalam. That means if I'm reading in this direction 3 prime to 5 prime, or if I'm reading in the opposite direction 5 prime to 3 prime, they are same. GAATTC, GAATTC. Can you see that 5 to 3, 5 to 3? It is the same. This is known as, yes, this is known as your palindromic sequence. What is the most important thing that the palindromic sequence do? The palindromic sequence are very important because restriction enzyme जो DNA को कट करेगी. जब तक DNA कट नहीं होगा, foreign DNA या foreign gene आकर attach नहीं होगी. So who's going to cut the DNA ताकि foreign gene आकर लगे? Restriction endonuclease. तो restriction endonuclease कहाँ से कट करता है? Restriction endonuclease palindrome को identify करता है और palindrome को ही कट करता है. Any doubts? Any doubts? Cool. Let us move ahead. Yes, so restriction palindromes are a group of letters that form the same word. For example, Malayalam, which is read in both directions. So the palindrome sequence is cut by the restriction endonuclease and cut होने के बाद गैप बन जाएगा. उस गैप में मैं foreign gene या जो भी मेरे काम का सामान है that I can add there. Yes. चलिए, moving on to the next question. Which of the following is not correctly matched for the organism as its cell wall degrading enzyme? इनमें से कौन सा correctly match नहीं है? Yes, very good, very good, very very well answered. Yes, everybody, such a simple question. Fungus का cell wall is made up of chitin, तो chitinase होगा. Bacteria का cell wall, this we have done at least 8 to 10 times. Bacteria का cell wall बना होगा NAG-NAM से. N-acetyl glucosamine, N-acetyl muramic acid. NAG-NAM से cell wall बनता है, which is degraded by lysozyme. प्लांट सेल वॉल बना होता है सेलुलोज का, जो डिग्रेड होता है सेलुलेज से. एल्गी में मिथाइलेज का कोई काम नहीं है. राइट। मिथाइलेज तो होस्ट सेल की प्रोटेक्शन के लिए होता है, कैपिंग में यूज करते हैं। सेल वॉल ऑफ एल्गी भी सेलुलोज पेक्टिन की बनी हुई है, तो इसके लिए भी हमें क्या चाहिए होगा, वी विल नीड सेलुलेज। मिथाइलेज इज अ टाइप ऑफ ट्रांसफरेज एंजाइम दैट ट्रांसफर्स अ मिथाइल ग्रुप। जहां पर भी मुझे मिथाइल ग्रुप लगाना है, फॉर एग्जाम्पल जब मेरा आरएनए बनकर तैयार हो जाता है तो मैं उसका कैपिंग कर देती हूँ। जब मेरा आरएनए बनकर तैयार हो जाता है मैं उसका कैपिंग कर देती हूँ। व्हेन द आरएनए इज रेडी आई एड अ कैप। The cap is added of 7-methyl guanosine. I add a cap ताकि मुझे पता चले ये रेडी आरएनए है, तो मैं क्या लगा दूंगी, एक कैप। The cap is made up of 7-methyl guanosine where I can use a methylase. Methylase is only used to transfer a methyl group. वो सेल वॉल डिग्रेडेशन से उसका कोई लेना देना नहीं है। करेक्ट। नेक्स्ट क्वेश्चन ऑन योर स्क्रीन प्लीज। एन एनालिसिस ऑफ क्रोमोसोमल डीएनए using the southern hybridization technique, right, does not use, when I have to analyze a DNA, whether this, you know, when I have to study the DNA in detail, I have to analyze the DNA, southern hybridize, PCR का कोई यूज नहीं होता। I have to analyze a DNA. I do a blotting. You know what is a probe? Let me tell you what is a probe. Suppose करिए, very important analysis of DNA. Suppose करिए मुझे DNA में AUG चाहिए. It's a very long DNA. Let me draw it for you. See, it's a very long DNA जहाँ कोने में एक छोटा सा AUG लगा है. I have to identify this part. So what I will do is very simple. I'm very smart. मैं UAC नाम का एक molecule बना दूंगी और इस UAC molecule पे मैं एक radio isotope लगा दूंगी. Pay attention. I will put a radioisotope so that everybody can see this, it will glow. तो ये UAC जाके AUG पे लग जाएगा obviously और यहाँ पे glow शुरू हो जाएगा because UAC लग गया है, रेडियो आइसोटोप है। So I have been able to, yes, I have been able to see it, right? So electrophoresis में I separate the DNA, blotting में I, देखो जो जो DNA के मेरे पास parts है, these are all part of DNA fingerprinting, right? Blotting में what I do is I take a nitrocellulose paper. जो मेरे पास DNA निकल कर तैयार हो गया, I need to put it on a piece of paper. Agarose gel electrophoresis, I separated the DNA. Now the separate pieces of the DNA, I repeat, agarose gel electrophoresis, I have separated the DNA. Now these separate pieces, I need to put them on a piece of paper and see कि ये डीएनए किस शेप के हैं, किस साइज के हैं, किस टेक्सचर के, दैट इज कॉल्ड ब्लॉटिंग। एंड फाइनली उनको मैं रेडियो आइसोटोप से लेबल कर देती हूँ। मैंने उनको पेपर पे लगा दिया। आई एम गोइंग टू लेबल देम विद रेडियो आइसोटोप एंड सी हाउ व्हाट काइंड ऑफ पैटर्न आर दे फॉलोइंग। आई रिपीट इट फॉर एवरीबडी। I separate the DNA with agarose gel electrophoresis. The separated parts, जो पार्ट मैंने agarose gel electrophoresis में सेपरेट किए हैं, जो पार्ट आगे गए हैं, जो स्मॉल पार्ट्स हैं, I need to put them on a paper which is known as nitrocellulose paper. So I blot them and then I fuse them with radioisotopes so that they form a pattern for me. Important. If you want this to be done in detail, let me know. I will do it. PCR is only for amplification. इसका hybridization, DNA fingerprinting वगैरह से कोई लेना देना नहीं है directly. Cool? It's not meant for southern hybridization. Which vector can clone only a small? See, vectors are of various types. There are vectors known as yeast artificial chromosome. There are vectors known as bacterial artificial chromosome. There are vectors also known as the human artificial chromosome. Each one of them can carry very large material. ये बहुत heavy load carry कर सकते हैं. But question ये है कि which one of the following जो mention किए गए हैं, आधा तो मैंने आंसर कर दिया, can carry only a small fragment of DNA. बताइए, which can carry? Yes, something has gone into my eye. That's okay, happens. Yes, exactly. Plasmid is meant for carrying small loads. It's meant for carrying small loads. Is that clear? Plasmids are extra chromosomal, self-replicating. मैंने आपको बताया DNA. देखिए ये इसका क्रोमोसोम है. Let me write with a black pen. ये इसका क्रोमोसोम है. ये दिख रहा है आपको प्लास्मिड्स, दे आर एक्स्ट्रा क्रोमोसोमल और बहुत स्मॉल मॉलिक्यूल्स होते हैं, ये अपने आप रेप्लिकेट करते हैं। इनके अंदर मैं फॉरेन सब्सटेंस डाल देती हूँ, यस, इनके अंदर आई एड अ फॉरेन सब्सटेंस, ये अपने आप रेप्लिकेट करते हैं। बच्चों प्लीज यूट्यूब लेक्चर देखिए प्लास्मिड का, मैंने ये पूरे का पूरा समझाया है। इट इज वेरी 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 इंपॉर्टेंट। एक बार दोबारा समझाती हूँ। देखो प्लास्मिड का काम है रेप्लिकेट करना, मतलब जो भी फॉरेन सब्सटेंस मैंने डाला है, इंसर्ट किया, देखिए ये इंसर्टेड जीन है, हो सकता है ये इंसुलिन की जीन हो सकता है ग्लूकागॉन की जीन हो सकता है किसी एंटीबॉडी की जीन हो, इसको इसने बार बार रेप्लिकेट करना है। सो एंटीबायोटिक रेजिस्टेंस जीन होगा विच इज अ मार्कर, एंटीबायोटिक रेजिस्टेंस जीन ही हमें बताएगा भले वो ट्रांसफॉर्मेंट है या नॉन ट्रांसफॉर्मेंट है। ओरिजिन ऑफ रेप्लिकेशन होगा, ये फटाफट फटाफट रेप्लिकेट करवाएगा। सिलेक्टेबल मार्कर, promoter, right, which will cause high number of copies, correct? Now the DNA जो वेक्टर होता है ये प्लास्मिड है, दिस इज अ कैरियर, दिस इज अ वेक्टर, तो अलग अलग तरह के वेक्टर्स हैं, राइट? Plasmids have been modified to be used as vectors. They can clone DNA fragments up to 10 kilobase size. Plasmids. Cosmids can carry how much foreign DNA? 45 kilobases. Yeast artificial chromosome. देखिए पिछली क्लास में मैंने आपको बोला है, ये जो ऊपर है YAC, राइट, ईस्ट आर्टिफिशियल क्रोमोसोम, बैक्टीरियल आर्टिफिशियल क्रोमोसोम, ह्यूमन, ये बहुत लार्ज अमाउंट ऑफ डेटा कैरी कर सकते हैं। सो YAC कैन कैरी दिस मच, BAC can carry this much, 300 to 350 kilobase pairs, 1000 to 25 kilobase, 2500 kilobase pairs by YAC। तो ये बहुत लंबा डीएनए फ्रैगमेंट, बहुत एक्स्ट्रा डीएनए फ्रैगमेंट कैरी कर सकते हैं, right? The DNA molecule, such a simple question to which the gene of interest is integrated. पिछले क्वेश्चन में एक फोटो दिखाई है, ये मॉलिक्यूल जिसके अंदर मैंने जीन ऑफ इंटरेस्ट डाल दिया है, what is this molecule called? It is obviously called a vector. It can be a plasmid, it can be a cosmid, it can be bacterial artificial chromosome, yeast artificial chromosome, human artificial chromosome. Yes, तो वो मटेरियल जिसके अंदर मैंने फॉरेन जीन डाल दी है is called a vector. वेक्टर, जब एक वेक्टर एक फॉरेन डीएनए ले लेता है, इट टेक्स इन अ फॉरेन डीएनए, इट बिकम्स अ रिकॉम्बिनेंट डीएनए। सो व्हेन द वेक्टर टेक्स अ फॉरेन डीएनए, इट बिकम्स अ रिकॉम्बिनेंट डीएनए। वो जॉइन कैसे होगा? हाउ विल आई जॉइन द वेक्टर प्लस फॉरेन डीएनए? द वेक्टर प्लस फॉरेन डीएनए इज जॉइंड विद द हेल्प ऑफ एन एंजाइम नोन एज डीएनए लाइगेज। इज दैट क्लियर? यस। एंड हाउ विल आई कट इट? फ्रॉम वेयर? फर्स्ट ऑफ ऑल वेक्टर को मैं कट कहां से करूंगी? वेक्टर में होगा रिस्ट्रिक्शन साइट, which is nothing but a palindrome. So the vector needs to be cut from the restriction site. What is the restriction site? Palindrome, GAATTC. Cut कौन करेगा? Restriction endonuclease enzyme cut करेगी. Then I will join it with the help of a DNA ligase. It is very, very simple. Is that clear? Perfect. Moving ahead to the next question. Which one of the following is not? Yes, एग्जैक्टली मैंने पहली स्लाइड में रिपीट किया था, वेक्टर ऑलवेज, प्लास्मिड्स ऑलवेज हैव टू बी, यस, दे आर ऑलवेज डबल स्ट्रैंडेड, दे आर नेवर सिंगल स्ट्रैंडेड। ट्रांसफर हो सकते हैं एक से दूसरे सेल में, यस। अपने आप रेप्लिकेट करते हैं, आर दे डिपेंडेंट ऑन द होस्ट? नो, दे रेप्लिकेट ऑन देयर ओन, दे हैव सर्कुलर स्ट्रक्चर बट दे आर ऑलवेज डबल स्ट्रैंडेड, दे आर नॉट सिंगल स्ट्रैंडेड। कूल? एनीथिंग दैट यू हैव नॉट बीन एबल टू अंडरस्टैंड? Are you all understanding? Plasmids are extrachromosomal, self-replicating, usually circular, double-stranded DNA molecules that serve as vectors which carry foreign DNA segment and replicate inside the host cell. Yes? Nice. Which one of the following is a restriction endonuclease? Restriction endonuclease are special enzymes, absolutely right, my dear children, which cut the plasmid. So these are enzymes which cut the plasmid. Which one of the following are restriction endonuclease? Yes, EcoR1 will cut it. Hint 2, Haemophilus influenza is a bacteria. Haemophilus influenza. So हमने Haemophilus influenza से बनाया है. Hint 2 is the first restriction endonuclease, direct MCQ. It is isolated from a bacteria by the name of Haemophilus influenza. RD is the strain. Right? It is the second enzyme to be isolated. Right? DNase, it is an endonuclease. It cuts the DNA from the middle. जो enzyme protein digest करे, protease. जो enzyme RNA को digest करे, RNase. जो enzyme DNA को digest करे, ये DNA है. जो enzyme DNA को end से cut करती है, we call it the exonuclease. जो enzyme DNA को बीच से cut करती है, we call it the endonuclease. Is that clear? Right? So Haemophilus influenza RD strain से लिया गया है hint 2. Haemophilus influenza RD strain. Correct? Protease enzyme proteolysis करेगी. Right? जो RNAs new RNA को cut करेगी into smaller segments. Right? And the DNAs or the RNAs can be exo or endo. एंडो आरएनएज या एंडो राइबोन्यूक्लियस आरएनए को बीच से कट करेगी. एंडो या एक्सो राइबोन्यूक्लियस आर द टू टाइप्स. एक्सो राइबोन्यूक्लियस आरएनए को बाहर से कट करेगी, एंडो राइबोन्यूक्लियस आरएनए को Exactly, बीच से कट करेगी. Any doubts? Stir tank bioreactors. Similar kind of question getting repeated again. What is the function of stir tank bioreactors? Absolutely right. वहां पे मैंने explanation में ये आपको बताया था. Stir tank bioreactors will make sure that everything is mixed with everything and availability of oxygen throughout the process. They are bioreactors जिनके बीच में एक stirrer है. They will move it so that oxygen is made available to the entire system throughout. Cool. A stirred tank reactor is usually cylindrical. It usually has a curved base. देखिए, yes, ये है stirrer and this will also release oxygen. Stirrer दो काम करेगा, सब कुछ अच्छे से mix करेगा and oxygen availability throughout the bioreactor so that it can move and make oxygen available. Any doubts? Any doubts? Cool. Easy questions. Easy questions. If you know it, you've been able to answer. One more question on agarose gel electrophoresis. कैसे दिखाई देंगे? I did it in the first question. Ethidium bromide. Correct. ब्रोमोफेनॉल ब्लू, एनिलिन ब्लू नहीं है. इथिडियम ब्रोमाइड यूवी रेज के नीचे डालिए, ऑरेंज कलर का सामान दिखाई देगा. करेक्ट. Put it under orange colored, put it under the UV light, you will be able to see. When you put ethidium bromide on agarose gel and you expose it to ultraviolet radiation, you will be able to see the DNA as bright orange. This has come in MCQs at least 20 times. Very, very simple, very, very important question. DNA fragments are agarose gel electrophoresis. They move towards this. They move towards the anode, which is positively charged. So DNA fragments have to be negative, neutral, positively charged, either positive. No, DNA fragments because of H3PO4. See, if you know it, you are able to answer it. Yes, and you feel happy after answering. कि आता है, 4 डालेंगे. Cool. That's the spirit of all my Vedans. Vedans have to rock it. Yes, everybody. High five. Cool. Perfect. Chaliye. Right? So DNA is negatively charged, so it has to move towards a positively charged anode. Which is the criteria? What is the criteria for DNA fragments movement on agarose gel? Yes, the smaller the fragment size, the less the molecular weight, the farther it moves. The negatively charged, these are the two fundas. The negatively charged DNA move towards the positively charged anode, funda number 1. And the smaller the size, जितना छोटा होगा. Yes, आप दो लोगों की रेस करा लीजिए, एक सेक्स है दोनों लोगों का, एक एज है दोनों लोगों की, एक थोड़ा सा बहुत हेवी ऑर्गेनिज्म है, राइट, लिटिल फैट ऑर्गेनिज्म, लिटिल स्लिम ट्रिम एथलेटिक ऑर्गेनिज्म, कौन ज्यादा देर से पहुंचेगा और कौन जल्दी पहुंचेगा? जो पतला है, सुडोल है, एथलेटिक है, वो दैट पर्सन विल रीच फर्स्ट ऑन द फिनिश लाइन. एंड जो मोटा है, unfortunately because of some health issue, that person is not able to maintain the body weight, that person will not be able to, yes, move as much. Is that clear? Electrophoresis is a technique used for the separation of substances of different ionic properties. Since the DNA fragments are negatively charged molecules, one last time I'm repeating everything, they will move towards a positively charged anode, right? The smaller fragments are less in weight, they're small in size, so they will move farther away. आप यहाँ सारा DNA डालिए, जो सबसे छोटा होगा वो सबसे पहले इधर पहुंचा होगा एनोड की तरफ. Small size, less weight, faster movement. Is that clear? Genetic engineering is, what is genetic engineering? Come on. Exactly. Hybridization of DNA of one organism to that of the others. Right? I take a plasmid from E. coli. I mix it with insulin gene of humans. क्या मैंने मिक्स कर दिया दोनों को? Have I taken E. coli plasmid and have I mixed it with insulin gene of humans? Absolutely. So have I hybridized DNA of one organism to that of others? Exactly. That is known as recombinant DNA technology, biotechnology, genetic engineering, hybridization of DNA of one organism. See, I'm removing it. Let's say this is human DNA. See, मान लीजिए ये इंसुलिन की जीन निकल रही है, मान लीजिए ये ग्लूकागॉन की जीन निकल रही है, and I'm removing them, और मैं किसमें ऐड कर दूंगी ये? Cool. Is that clear? Any doubts? Cool. In genetic engineering experiment, DNA fragments are joined by, हमने अभी किया है, गायरेज और हेलिकेज तो अनवाइंड करेंगी, टोपोआइसोमरेज, गायरेज ये डीएनए को रेप्लिकेशन के टाइम ओपन करेंगी, राइट? रेप्लिकेशन के टाइम दे विल अनवाइंड, दे विल ओपन. पॉलीमरेज तो डीएनए को सिंथेसाइज करवाएंगी. दे कैन बी आरएनए पॉलीमरेज और दे कैन बी डीएनए पॉलीमरेज. स्पॉट वेल्डिंग, दो पॉइंट्स को इमीडिएटली जोड़ देना. स्पॉट वेल्डिंग, there are 2 points I'm quickly wanting to join them. This is done by, yes, ligase. Is that clear? Right, so it is ligase. Ligase is the one which joins them. Recombinant DNA technology is the manipulation of DNA from different source so as to recombine the DNA portion. During this portion, the DNA are broken into segments using restriction endonuclease. After inserting the desired genome, the fragments are allowed to fuse with the help of DNA ligase. So एक जगह से आप एक fragment निकालिए, दूसरे से दूसरे. How will you fuse them? Yes, you will fuse them with the help of DNA ligase. It's a spot welder enzyme. It does welding of the DNA parts. Any doubts? Who discovered the recombinant DNA technology? Was it Mr. Hargobind Khurana, who was from India and studied in India for a long period of time? Was it Watson, Cohen, and Herbert Boyer in California, or was it Sutton and Lowry? Yes, it was Stanley Cohen and Herbert Boyer. Correct. Recombinant DNA technology or genetic engineering is the technique which changes the chemistry of genetic material, because इसमें हम कोई host organism के अंदर मैं कोई foreign DNA डाल दूंगी. The science of recombinant DNA technology took its birth when Cohen and Boyer in the year 1972 were able to introduce a piece of antibiotic resistance gene containing foreign DNA. Yes, foreign DNA इन्होंने किसके अंदर डाल दिया था? Salmonella typhi के अंदर. The modified plasmid was then inserted inside E. coli और E. coli के अंदर वो reproduce करने लग गया. Genetic engineering क्यों possible है? Is it possible because we can cut DNA at specific sites by endonuclease, like DNA is one? Restriction endonuclease purified from bacteria can be used in, can we cut DNA with DNA is one, or do we cut it with the help of restriction endonuclease? The phenomenon of transduction in bacteria, transduction in virus. We can see DNA, so we can do genetic engineering. No. What is the answer? Because we have restriction endonuclease which can cut the DNA at very, very specific points known as palindromes. ना रहेगा बांस ना बजेगी बांसुरी. There is no palindrome, there is no restriction endonuclease. We are not able to absolutely, yes, cut and fuse. और join कैसे करते हैं? Cut DNA को join कैसे करेंगे? With the help of an enzyme known as ligase. Correct? So DNA restriction endonuclease are a class of restriction endonuclease which cut the double-stranded DNA. रिस्ट्रिक्शन एंडोन्यूक्लियस को हमने बैक्टीरिया से आइसोलेट किया. ये बैक्टीरिया ने खुद बनाया है, ह्यूमंस ने नहीं बनाया है. वी जस्ट गेट इट फ्रॉम द बैक्टीरिया. फॉर एग्जांपल हिंटू, ये कट्स बना देते हैं, ये डीएनए को कट कर देते हैं. प्लीज सी द वेबिनार. मैंने स्टिकी एंड्स वगैरह सब कुछ डिटेल में समझाया. प्लीज 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 सी द यूट्यूब लेक्चर. राइट, नीट वेदांतु चैनल का यूट्यूब लेक्चर है. इट्स प्रेजेंट ऑन द नीट वेदांतु चैनल. इज दैट क्लियर? सो व्हेन आई कट द डीएनए, देखो व्हेन आई कट द डीएनए, ये था डीएनए पूरा, ये इसके हाइड्रोजन बॉन्ड्स. सो व्हेन आई कट इट, क्या हाइड्रोजन बॉन्ड सेपरेट हो गए? कैन यू सी दैट? यस, ये देखिए ऊपर नीचे. सो दे आर स्टिकी एंड्स, राइट, एंड आई सेपरेट देम लाइक. ये डीएनए था, so ऊपर के sticky ends and नीचे के sticky ends, and then I insert a foreign substance inside it. Is that clear? Right, so restriction endonuclease is able to cut it. Right, last question for the day. Interesting class, simple class, easy class. If you've read it, if you've not read it, you will not be able to answer it. If you've read it, you will be able to answer it. I will see you again tomorrow on biotechnology. By the time, I want you to see all the webinars and the YouTube lectures and come. The technique which involves addition or deletion of genes is, I am adding genes or I am deleting any gene, is known as genetic engineering. There is a disease which is known as severe combined immunodeficiency, where the person does not have energy, does not have immune cells. This happens because of the deficiency of ADA, adenosine deaminase की deficiency की वजह से होता है. इसके ट्रीटमेंट में हम एक अच्छी जीन बॉडी में डाल देते हैं जो एडीए बना सकता है. दिस इज नोन एज जीन थेरेपी. टू बी एबल टू कट द जीन्स इज नोन एज जीन स्प्लाइसिंग. Artificially synthesizing genes is known as artificial synthesis, but addition, deletion, modifications of gene is known as genetic engineering. Yes, so genetic engineering is a technology which involves synthesis of artificial genes, repair of genes through fusion, deletion, addition, production of recombinant DNA technology. We manipulate genes for the betterment of human beings, plants, animals, and microbes. So that, my dearest children, brings us to the end of another super cool, super amazing, extremely informative session on biotechnology, which we kind of did very, very quickly. Yes, because all of you know the theory in detail. All of you by this time know it. Now I will want to leave this session, but I want to leave it on two things which I want to tell you. Point number one, please message your doubts in comments. And point number two, please tell us in the last 20 days what all are the topics that we should revise. Should we make theory lectures? And do you want us to make theory lectures on which all topics? Please write it to us and then we will do the needful. किस किस टॉपिक्स पे आपको क्या क्या चाहिए, what all are the difficulties are you facing, please write it down in the comment section and the needful will be done. Yes, so that is Dr. Vani signing off for today on behalf of Vedantu. I hope you all the success that you ever dream of. I hope you achieve all your dreams. I hope you study very, very hard. God bless you, everybody. Take very good care of yourself. Eat well, sleep well, and study very, very well. Bye, children. Take care. See you tomorrow, same time, same place.